Okay, welcome to the first session of the ASBMB Transcription Webinars. The monthly transcription webinars aim to promote collaboration and knowledge sharing among scientists interested in the general areas of transcription, chromatin, and epigenetics. Today we have two truly outstanding speakers. Each speaker will talk for about 25 minutes, followed by answering questions for about 5 minutes. So when the audience asks questions, please raise your hand. You may also ask questions in the chat box. After both talks, I will open 2 breakout rooms, one for each speaker to answer additional questions for 10 to 20 minutes. And today, Dr. Alyssa Lei, a senior investigator at NIH, will also co-host together with me. Our first speaker is Professor Rob Klose from Oxford University. And the title of his presentation is Understanding How CpG Islands Control Transcription. Welcome, Rob.
Thanks very much, Kai. I'm just going to hopefully share my screen.
Yeah, it's right.
It's right. Yes. Is it presenter or mode or correct?
It's correct.
Fantastic. Okay, great. So first of all, I just want to thank Kai for keeping this seminar series going. I think it's been really fantastic over the past years. And it's great to be able to be part of the first one in its new format. So thank you to Kai and the other organizers. And I get the honor of being Ken's warm-up act today, so I'm really looking forward to that as well. All right. Okay. So what I want to share with you today is some work that we do in my group trying to understand how CpG islands regulate the process of transcription. And I think probably for this group, we can probably all agree at its most simplistic level that the ability to control gene transcription relies on DNA binding transcription factors engaging with the regulatory sites in the genome and helping to control the way that RNA polymerase II ultimately engages with the promoter and drives transcription. But interestingly, what we've learned through studying the process of transcription in eukaryotes is that the template on which transcription occurs can also profoundly affect gene expression. So for example, nucleosomes can occlude the way that RNA polymerase or transcription factors engage with DNA, but also you can add post-translational modification to the tails in histones that make up nucleosomes, and these can also really impact the way genes are used. And really that's been the general focus of the work in my lab, trying to understand how chromatin environments around gene promoters regulate gene transcription. And we largely do that through studying an interesting class of vertebrate regulatory elements that are called CpG islands. So these are short contiguous regions of the genome that completely lack DNA methylation, and they're associated with the vast majority of vertebrate gene promoters. So for a long time, we had no idea what CpG islands were actually doing. And I think a pretty important discovery came a number of years ago when we and others found that there's a DNA binding domain called a zinc finger CXXE domain that can specifically recognize non-methylated DNA in CpG islands. So it's effectively a reader of where these domains are in the genome. And when we went in and purified the proteins that had the CXXE domain, what we found is that they were all part of large chromatin modifying complexes. So this sort of indicated that the ability to read where islands was in the genome was helping to bring in chromatin modifying systems to gene promoters. And this was sort of a little bit of a different idea because we'd often thought about transcription factors binding specific sequences and bringing in these enzymatic complexes. But here we were showing that a domain that recognizes virtually all promoters can bring in these activities. So this immediately gets you thinking about, well, what is it then that defines how distinct chromatin states are ultimately set up at gene promoters? And I'm not going to go into the details of this, but work from us and others has shown that once these complexes bind, they have an ability to sort of sense the underlying transcription state of the gene promoter. So if a gene is off, a subset of these CXXE domain complexes can recognize this and install repressive histone modifications that then help to maintain the inactive state. Whereas if a gene is being actively transcribed, there's a distinct set of complexes that sense this. They install histone modifications that are thought to be permissive to transcription, and thus they help to maintain gene expression. So we've worked a lot on both of these distinct activities. The more repressive systems you'll know as the polycomb repressive system. And if you want to look at some of our more recent work, you can look at Alex's paper from a couple of years ago. But today what I want to focus on is this alternative set of complexes that drive this more permissive state. And these fall into the trithorax group of chromatin modifiers. So what are these? Well, the ones that we're going to be considering today are the SET1 and MLL histone lysine methyltransferases. So they have one of these CXXE domains, and what they're able to do is localize to CPG islands and use their histone methyltransferase activity to add either 1, 2, or 3 methyl groups to the tail of histone H3 at position 4. Important to the functionality of these systems is actually the ability to associate with this complex called the WRAD complex. And what it does is it couples with the set domain of these SET1 MLL proteins to create the enzymatically active core. Probably the most studied group or member of this family are the SET1 A and B proteins. They have one of these zinc finger CXXE domains, and they tend to be targeted to CPG islands and drive histone H3 lysine 4 trimethylation. And they seem to do so, be particularly important for this modification at highly transcribed or actively transcribed genes. Now, in contrast, the MLL1 and 2 proteins also have one of these zinc finger CXXE domains, but they tend to work at more moderately to lowly transcribed genes, again, driving trimethyl histone H3 lysine 4. So there's 2 additional members of this family called MLL 3 and 4. They also associate with these WRAD proteins, but they lack one of these CPG island targeting DNA binding domains. And instead, they're thought to localize primarily to enhancers through interacting with transcription factors. And actually, Kaya has done a lot of the really excellent work characterizing these important MLL3-4 complexes. But what I want to focus on here today is the SET1 and MLL1-2 complexes, because they're the guys that recognize CPG islands and primarily are doing trimethylation of K4 at gene promoters. Okay, so what we know about the functionality of these systems can probably be boiled down into a very simplistic model whereby the SET1AB complexes, for example, can combine to CPG islands, combine to these CPG island elements. And if a gene is being actively transcribed, they can actually associate with RNA polymerase II, particularly when it's in its initiating form. And this seems to stabilize these complexes and help to drive up trimethyl K4 levels. It's then thought that reader proteins can come along and associate with trimethylation on K4, and that these then more directly affect the process of transcription. And that this process would be important then in creating a sort of feedback loop whereby transcription helps to drive SET1 recruitment, which helps to police K4 and drive more transcription. And so these simplistic models are really based on a series of rather disparate observations. And therefore, in many cases, it's actually unclear whether these sort of ideas can explain the functionality of these systems inside cells. And indeed, if you look carefully at the literature, I think there's a series of questions which, despite the fact that we've known about these complexes for decades, still remain very poorly understood. So the first of these is really how do these grouping of SET1 MLL methyltransferases actually shape trimethyl K4 throughout the genome, and how is that then related to transcription? And also so probably the reason that that's been difficult to pin down is the multiplicity within this system. So there's at least 6 of these histone H3 lysine 4 methyltransferases. So removing these individually or in combination has been a real technical challenge. And the second of these questions is really once these sort of chromatin states are set up at a gene promoter, then how mechanistically do they ultimately control the process of gene transcription? And so the reason it's been difficult to understand these sorts of mechanisms is usually the way we try and approach these problems. So often we'll take wild type cells and we'll make knockouts or use short hairpin RNAs to take away one or maybe a few of these complexes, isolate these knockout or knockdown cells, and then try and look at transcription. Now, the problem is that many of these complexes are very important for transcription. So you take them away, you alter cell fate, and it's very difficult to know whether you're looking at secondary effects or the primary effects that these systems have on transcription. So a number of years ago, we reasoned that if we were going to understand these systems, we really needed to think of alternative ways that we could try and capture these really sort of primary effects. And so we thought what we needed is systems where we could rapidly disrupt these systems and then be able to capture the most primary effects that they have both on K4 methylation, transcription, and the underlying mechanisms of transcription. And so the way that we set about to do that is to use degron depletion strategies, which many of you will be familiar with now, but for those of you who aren't, what we do is we engineer small tags into the endogenous genes for coding these proteins. And what this which will target an E3 to your protein of interest, it will polyubiquitinate it, drive it to the proteasome, and turn it over very, very rapidly. So we brought to bear this sort of degron technology on the SET1 MLL complexes, and we first focused on SET1AMB because it was thought to be really the prominent K4 methyltransferase working at active genes. So we generated a degron cell line that allowed us to simultaneously remove SET1A and B. And then what we did is we depleted it and carried out chip sequencing for histone H3 lysine 4 methylation. And we analyzed this at 2 hours and 8 hours after removal. And I think what you can see from this meta plot of all gene promoters is actually the effects we observed were very, very modest. Okay, there are effects, but they're very small. And so this sort of surprised us because we thought these should be, based on much in the literature, the major methyltransferases, certainly at active genes. So this led us to think that, well, maybe some of these other complexes were contributing. And the obvious targets would be MLL1 and 2, which also have CXXE domains and go to gene promoters with CpG. So we generated degrons where we could remove MLL1 and 2 simultaneously and carried out the same analysis. And now we actually saw much more dramatic effects on K4 methylation, probably about a 40 to 50% reduction. So this tells us, I think, that MLL1 and 2 together are actually having quite a broad effect on K4 methylation throughout gene promoters in the genome. But of course, when you look at these two plots, you'd say, well, it doesn't seem to explain the K4 methylation that you're seeing in wild type. So we wondered whether maybe SET1 and MLL1-2 complexes were working together at these sites and there was overlapping function. So we then generated a degron cell line where we could remove all 4 of these complexes simultaneously and again carried out ChIP-seq analysis. And I hope what you can see now is these effects are much larger, probably about a 60% or more reduction in trimethyl K4. So this tells us that these complexes are indeed working together, and in some instances, they appear to actually be synergizing with each other to drive these effects. But of course, if you look at this, you can still see there's quite significant levels of trimethyl K4 left. So this led us to believe that there may be some alternative trimethyl transferase activity that we were missing. So we began to wonder whether these additional methyltransferases, MLL3 and 4, might also play a role in driving tri-K4 to these sites. So we built a degron line where we could remove all 6 complexes simultaneously and did the same analysis. And now what we saw was much larger effects on trimethyl K4 with a 75% or greater reduction. So I think this tells us that actually it's a culmination of these complexes that are ultimately driving the observed trimethyl K4. But interestingly, when we looked beyond 2 hours, we saw something we didn't expect, which is that the K4 trimethylation levels actually seemed to increase. So this made us wonder whether we might be missing some undiscovered or uncharacterized methyltransferase activity that was now compensating in the absence of these canonical complexes. So we wanted to try and determine whether that was indeed the case. So the first protocol was just to make sure that we really were removing the SET1 MLL complexes. So just to remind you, we have these 6 different complexes. And if you analyze them biochemically by size exclusion chromatography, what you can see is they all run as very large molecular weight complexes. And if you look at the shared components of the WRAD complex that make them up, you can see that these proteins co-migrate with these regions in the exclusion matrix. And this is important because these are small proteins, so they're being dragged into these large molecular weight complexes. So we believe these are the native SET1 MLL complexes. So we then used this assay and carried out degron depletion to remove these 6 proteins. And you can see very nicely that if you Western fold them, they're now completely gone. So we are efficiently depleting them. And importantly, if you look at their WRAD components, they now collapse down to these low molecular weight fractions. So the complexes are indeed being broken up. But what we found very interesting is there appeared to be some high molecular weight complexes that were retained that had these WRAD components. And when we looked for other factors that might co-migrate, we found that CFP1, which is usually part of these SET1 complexes, was also migrating in these high molecular weight fractions. So this suggested to us there may be an alternative WRAD CFP1 containing complex in these cells, and that it might have a novel set domain contributing to K4 methylation. So to try and determine what this might be, we carried out biochemical analysis and immunoaffinity purified the core WRAD complex, either in a wild type scenario or after removing all 6 of these methyltransferases. And what this allows us to do by comparing these data is to look at proteins that are lost from the complex, maybe those that are increased or those that are retained. So importantly, we saw none that were increased. Importantly, we saw the things that we had Degron tags on were being depleted, and we retained the core WRAD components because, of course, they can still associate even without their scaffold for these larger complexes. But what really surprised us is we also saw that CFP1 and the SET1B protein were retained in these purifications. Now, this was completely nonsensical to us because we Degron tagged SET1B, so it should be completely gone. So what we did is we went in and we analyzed where these peptides were coming from in this complex. And what we found is that they almost all corresponded to the C-terminal region of the SET1B protein, okay? Suggesting there may be an unidentified shorter isoform of SET1B that we didn't really know about. And indeed, when we use an antibody against the C-terminus of the SET1B protein, you see the full-length protein here that is removed by Degron addition, but this shorter C-terminal fragment doesn't seem to be. Importantly, this associates with WRAD and is not dependent on the full-length protein. And I'm not going to bore you with all the details, but we went in and looked, and there's an alternative promoter within this gene that actually creates a new start site and a new short form of SET1B. So although I told you at the beginning that there were 6 methyltransferase complexes, we now discover there's a novel SET1B C-terminal complex that may then also contribute to K4 methylation. So we then generated a Degron cell line where we could remove all 7 of these complexes simultaneously and carried out lysine 4 trimethylation analysis again. And what you can see now is much more profound effects on K4 trimethylation. And in fact, we see very, very large effects when we look across the genome in toto. So we wanted to be absolutely sure that we were indeed now getting rid of all the WRAD SET1 MLL dependent activities. And so to ensure this was the case, we did a sanity check and we built a Degron where we could remove RBP5, which should inactivate all of these complexes, and carried out the same analysis. And what we see is effectively exactly what we get when we remove all 7 of the set domain containing proteins. So we're pretty confident now that we've got the vast majority or all of the activity that is present in these cells. Another way we validated this was to do ChIP-seq for these factors after removing SET1 and MLL, and those proteins are also lost from chromatin. So we were then also keen to understand, well, what might happen to the SET1 and MLL proteins when we depleted the WRAD components? So what we did is we carried out ChIP-seq for SET1 MLL proteins after RBP5 depletion. And we got a bit of a surprise here again, because what we saw when we looked is that actually, if you remove WRAD, these proteins still localize to their appropriate target genes in the genome. And if you look throughout the genome, if anything, their levels actually go up a little bit. So I think this tells us you can remove SET1 MLL, get rid of you can remove WRAD, get rid of trimethyl K4, but these SET1 MLL complexes will still localize appropriately. So we realized that this actually created a really unique opportunity for us because we had the ability to remove SET1 MLL proteins, to wipe out trimethyl K4, and to lose occupancy of these complexes in toto. But when we remove RBP5, we simply wipe out trimethyl K4 and leave occupancy of SET1 MLL complexes intact. So what we can do is now, by carrying out transcription analysis, understand the contribution of lysine 4 methylation and the occupancy of the complexes themselves to any effects on transcription. And the way we do this is to use TT-seq analysis, which is a nascent transcription analysis. And what we do is we pulse label these cells for a short period of time with 4-thiouridine. We then isolate total RNA and purify this very nascent RNA, and we carry out differential transcription analysis. Okay, so that's what we've done. And we're going to start by looking at these SET1 MLL proteins. So we deplete all 7 of those complexes. As I've shown you, this leads to a major reduction in trimethyl K4. And if we look at this target gene, what we now see is that transcription almost completely collapses. And if we look across the entirety of the genome, what we see is over a quarter of genes are absolutely relying on SET1 MLL complexes for their transcription. Okay, so now we can do the other experiment where we remove RBP5. It depletes trimethyl-K4 almost completely again. But now if we look at this target gene, we see transcription is completely unaffected. And in fact, when we look across the entirety of the genome, we see virtually no effect when you just remove trimethyl-K4. So if we come back to the question that I asked at the beginning, then how do set1 complexes define trimethyl-K4, and is this required for their effects on transcription? What we observe is that the set1 MLL complexes work together to shape trimethyl-K4 promoters and CPG islands. If you disrupt the set1 MLL complex components at their core, you see a loss of trimethyl-K4 and really profound effects on gene transcription. But if you simply remove RBP5, wipe out trimethyl-K4, and leave the complexes on chromatin, there's very little effect on transcription. So we think this indicates that the set1 MLL complexes themselves may be playing a really important role in driving transcription of its target genes. So this then opens up the ability for us to begin dissecting how these set1 complexes and MLL complexes mechanistically control the process of transcription. Now, there's a number of ways that we can do this. So for example, we could go in and use our Degron cell lines, deplete our proteins of interest, and carry out various different genomic analysis, maybe looking at transcription related factors, GTFs, other things. But the problem with these experiments is you're grinding up millions of cells. So you're effectively looking ensemble at what is happening to transcription. Okay, so this is all right, but what it really tells you is about average trends across the cell population. So you can determine whether things might be going up or things might be going down. But this becomes problematic when we start to think about transcription as it happens from the gene promoter. Because what we now know from live cell transcription imaging is that if you look over time at individual cells and individual genes, their transcription is actually highly stochastic, and pulsatile. So what you'll see is the promoter will be in an off state, it'll enter into an on state, initiate a burst of transcription where multiple polymerases enter into initiation and elongation, and this will happen very stochastically. But the really key thing is if you look at this process in maybe 2 or 3 other cells in the population, the same behavior will happen, but it'll be happening at different places and different times in different cells. So ensemble analysis means you average this all out. And this is really problematic because we now know transcriptional control relies on modulating the size, the duration, the frequency of these transcriptional events. Okay, so we reasoned if we were going to be able to understand what these complexes were doing to transcription, we needed to be able to look at transcription in single cells. So we took advantage of really well-developed technology, which is based on the MS2 repeat system. So here you put these repeats into the intron of your gene of interest. Polymerase moves across it when it's transcribed and produces aptamers that can be bound by a GFP-labeled protein. This means that when you see a focus of fluorescence, that corresponds to transcriptional events. And what you can do is create videos that, if it'll play, maybe not, will allow you to watch this pulsatile transcription over time and actually be able to record movies and quantify these transcriptional behaviors. So from this, what you can begin to do in many, many cells is extract information about the duration of these bursting events, their amplitude when they're on, their duration, and other kinetic behaviors. So what we did is we applied that technology to look at a set1 MLL target gene called PANK2. Along the x-axis here, we have 4-hour imaging movies. The y-axis is hundreds of cells, and the heat map shows those bursts of transcription. Now, when we remove the set1 MLL complexes, what you can see, hopefully from this heat map, is transcription is really dramatically affected. And if we do the same for removing just RBP5 and leaving those complexes on chromatin, there's virtually no effect at all. So what we use is these transcription heat maps and trajectories to then begin to quantify these behaviors. So we first started by looking at these on period when there's bursts of transcription and quantifying these, because this is when transcription, of course, is actually happening. So we first looked at the amplitude of these bursts, and what you can see when you remove set1 MLL is the amplitude of those bursts go down. And if you look at the on period or their duration, what you can see is, again, they're also reduced. So if we abstract this to this cartoon here, what we think is going on is that when you remove set1 MLL complexes, these burst sizes are actually smaller. So polymerases are either being lost or reduced in some way within the burst itself. Now, there's a number of different ways that this could manifest. One could be mechanistically from a reduction in the transcription initiation rate within a burst, or possibly termination of the burst or components within the burst once they've initiated. So we wanted to try and determine which these two might be the most likely explanation. And we already thought it might be termination because we published work a little while ago which had actually shown genetically that there was an interaction between SET1-MLL and a premature termination complex called the restrictor. So we wondered whether this might be counteracting restrictor's activity. So we went back to our system where we removed SET1-MLL and we engineered those degrons so we could simultaneously remove restrictor. And I think quite remarkably, you can see now this completely restores normal transcriptional activity. And if we can go in and quantify this in exactly the way I just showed you a minute ago, when you remove the SET1-MLL proteins, we have these reduced amplitude of bursts. Now, if you also co-deplete restrictor, it restores it. And in fact, it even goes a little higher, suggesting SET1 can't fully counteract this. And if we go in and we look at the duration of these, again, we nearly completely rescue it. So what we're seeing is a near complete rescue of these burst size by removing this termination mechanism. So we think primarily the SET1 complexes are actually blocking the activity of restrictor in order to maintain transcription burst size. Okay, so I think I'm probably coming towards the end here. I haven't been keeping track of time, but I'm just going to wrap up and just give you a recap of what I've said to you. We started with two very simple questions. How do these SET1-MLL complexes define K4, and how is this related to transcription? And how do these complexes mechanistically control transcription? So hopefully, I've been able to show you if you remove RBP5 and wipe out trimethyl K4, you have very modest, if any, effects on transcription. Whereas if you remove SET1-MLL, you wipe out trimethyl K4, but you also have really profound effects on transcription. In thinking about how this happens mechanistically, it looks to be a direct effect of these complexes themselves. And what we find is that they're important for maintaining the burst size. Without SET1-MLL, you get restrictor, seemingly picking off individual polymerases within bursts. But if you remove the two of them simultaneously, you can completely restore this. So we think this is good evidence that by localizing to CPG islands, the SET1-MLL complex is able to ensure that these genes can block restrictor activity and ensure that they're actively transcribed. And just one very quick final thought, we think this might be important for genic transcription because restrictor can work everywhere in the genome to terminate inappropriate transcription. So maybe CPG islands help to mark promoters to ensure you don't get inappropriate premature transcription termination, thus making them important for gene expression. Okay, very quickly, the important people who did this work, this nonchalant-looking chap here is called Alan. He's the brave soul that generated all these degron lines and did this beautiful work. He worked closely with Alex on the transcription imaging, and we had help from our friends Mikhail and Inga in doing the mass spec analysis. And all the work in our lab is funded by the Wellcome Trust. So thank you very much, and I'm sorry if I went a bit over time.
Thank you, Rob. Great talk, great work. We have time for 2 or 3 questions. So if the audience have questions, please raise your hand. Okay, Michael.
Oh yeah, thank you. Great talk, Rob. Really fascinating. I'm wondering, especially regarding the Degron system with RBBB5, was that you said that it has a big effect on HK4 trimethyls with little effect on transcription, but was that based on global transcription, or is there a more gene-specific effect that's sort of hidden in there by doing the bulk sequencing?
Yeah, so I went through it very quickly. I apologize. The TT-seq analysis is looking at all genes, so all 20,000 genes, and there's about 100 and something genes go down and about 200 genes go up. The effects are very, very modest, and there doesn't seem to be anything obvious about those genes.
Okay. All right, thanks.
Okay, Jackson.
Hi, Rob. That was great. Just really beautiful work.
Thank you.
I was wondering whether you see any difference in promoter TSS acetylation when you either deplete RBBB5 versus all of the methylases.
Yeah, I'm afraid I don't think we've done that carefully. I'd have to ask Alan whether he's ever looked at that. I mean, my expectation would be probably, because when other people have looked in similar contexts, they did see an effect, but I can't say because we haven't done it ourselves.
Thanks.
Okay, final question from Gaoya Man.
Hi, Rob. This is really cool talk. So I just wonder, I really like the idea of the set1 complex interact with the restrictor, although I don't know how that happens, but you I think so for H3K4 trimethylation, set1 complex, to my understanding, they all localize to promoter area, whereas for a long gene, termination happens to the very end of the gene. How do you think that would
Yeah,
uh yeah,
yeah,
usually
yeah.
sorry, usually I would think I've seen like H3K4 monomethylation being deposited at the end of a gene, but not like H3K4 trimethylation. How do you think of that?
So this restrictor complex is very special in that it works promoter proximal. So it works sort of within about the first 500 to 1 kilobase of a gene promoter, which actually corresponds to precisely where the set1 complex binds. So it is promoter proximal. The sort of termination complexes you're thinking about, the cleavage and polydenylation
Hmm.
specificity factors, which work at the 3' end. So yeah, I'm sorry, I didn't have time to really go into that, but it's a promoter proximal premature termination system.
Okay, thank you for answering that.
Thank you.
We have additional questions, but we have to move on to the next speaker. And please come to the breakout room to ask Rob more questions. Okay. So next, Alyssa Lei will introduce the second speaker.
So our next speaker needs no real introductions. Ken Zaret from UPenn School of Medicine. He's going to be talking about overcoming chromatin barriers to change cell fate.
Okay, so I want to thank Kai, Alyssa, and the ASBMB for the opportunity to talk. It's great that we can be here today. What I'd like to discuss is my personal interest in how biology can be digital, so to speak. In other words, you can look at phage, bacteriophage. They have a lysis or lysogeny genetic programs for each. Bacteria, vegetation or sporulation, vegetative growth or sporulation, yeast, different mating types. And there are different genetic networks that are activated and the mutually negative ones repressed. And somehow biology winds up with mutually exclusive cell fates, even though we know that underlying all of that now, there's a lot of movement. Everything's dynamic. Things are forming and breaking down and so forth, but nonetheless, it comes together. I'm showing the names of the illustrious scientists who've studied this in the past. And so how do we think about controlling mutually exclusive cell fates in higher organisms like us, where liver genes are on in the liver, brain genes are off, et cetera, for the brain? And what's underlying a lot of the cell fate transitions that occur in embryonic development, reprogramming, regeneration, and even cancer is that to activate a silent network that would oppose the original fate, you have to overcome chromatin barriers. And we know from extensive work from ENCODE that there are three broad types of chromatin, if you will. There's open chromatin, and a few percent of the genome is hypersensitive, and that's where active genes are. There's low signal or naive chromatin that's bound by linker histones and transcriptionally silent, but not particularly enriched for other marks. And then there's straight up heterochromatin, which has H3K27, trimethyl, H3K9, di, and trimethyl, and so forth. And so that's the landscape. And what we discovered many years ago is that there's a subset of transcription factors that are among the very first to bind genes in embryonic development, like FOXA1 and GATA4. And these factors have the particular characteristic, biochemical characteristic, of being able to bind sites on nucleosomes better than proteins that are not the earlier gene binding factors in embryonic development. And so we reconstituted chromatin in vitro. This was done by Lisa Cirillo, compacted it with linker histone, and showed that those factors not only targeted their sites in compacted nucleosome arrays in the absence of remodelers, but they would lead to a local opening of the chromatin or hypersensitivity, which would be depicted by that red nucleosome in the center. So the question is, how do these work and what do they do? Well, time has passed. Jussi Taipale published a beautiful study in Nature quite a while back now, showing different ways that these transcription factors can bind to nucleosomes. It's not just one type. We also know it's not a quantal effect. Some bind better than others and better than others that don't bind at all. So there's a gradient of it, and that's what we're also trying to understand. In addition, what we've kind of discerned using biophysical techniques such as FRET and FRAP and single molecule tracking is that good nucleosome binding factors traverse in the chromatin in live cells more slowly than the weaker nucleosome binding factors like MYC, let's say, or HNF4. And so the hypothesis is that the latter proteins that don't bind nucleosomes well exchange with open DNA sites and diffuse through the nucleoplasm, whereas the nucleosome binding factors sort of crawl across on and off the chromatin by virtue of their nucleosome binding capacity, even their, as we have shown, their non-specific nucleosome binding capacity. Well, fast forward, and Yawen Bai and Bingwing Zhu have beautifully summarized a lot of work from many labs in a review last year now that describe in vitro studies using mostly biophysics to discern how transcription factors that bind nucleosomes can disrupt linker histone, thereby opening up the nucleosome, if you will, and exposing it in an array. And so there's various different ways that this can happen. So we have different ways that this class of factor can bind nucleosomes and different ways that they can perturb nucleosome structure. So putting this all together, we have sort of an ABC model where there's the initial nucleosome scanning and targeting. There's the ability to disrupt structure locally. And then finally, there's the recruitment of nucleosome remodelers to further open the region and bump off the nucleosome. And I should say that Toshi Tsukiyama and Carl Wu actually showed this C step over 30 years ago, looking at GAGA factor and NERF and reconstituting in Drosophila extracts the ability of GAGA to elicit a nucleosome disruption by NERF complex. My lab's kind of been focusing more on the transient initial events that occur where the transcription factors are scanning the genome and leading to a local nucleosome disruption. So let's put this in the context of cell fate changes. And so now we know that diverse transcription factors, shown in bold here, that can bind nucleosomes are all at the top of genetic hierarchies for controlling cell fates in animals. So these are bona fide gene network regulators. And perhaps not surprisingly, all of those shown are capable of reprogramming cell fate from human fibroblasts and mouse fibroblasts to neurons, macrophages, hepatic cells, and induced pluripotent stem cells. I should point out that Abdunor Sufi's lab showed a number of years ago by screening different mutations of the OCT4 DNA binding domain, they found a mutation that didn't affect DNA binding. It affected nucleosome binding. It perturbed it, and it greatly reduced the ability of the factor to reprogram cells to pluripotency. So that was a good demonstration of how nucleosome binding is required. So what we wanted to do is actually look at biophysical and cross-linking techniques and so forth to actually identify interactions with the nucleosome that might or might not allow the factor to reprogram cell fate. And so we were focusing now on ASCL1, and I'm going to talk about a new unpublished set of work. So Marius Wernig's lab made the discovery that ASCL1, in combination with two other factors, can reprogram fibroblasts to neurons, and he eventually showed that ASCL1 could do it alone. In my lab, we found that ASCL1 bound DNA poorly, but if you had co-expression with E12A, another B-HLH partner, you could get it to bind nucleus-free DNA very well. We also found that the ASCL1 in our hands was not great at reprogramming fibroblasts to neurons, but in fact, if you had the ASCL1 and E12A co-expressed, it reprogrammed neurons like a champ. So it worked really, really well. And then looking at the factor for how it would reprogram chromatin itself, when we're delivering both factors and do a chip for ASCL1, you can see by the first red arrow that we get 18,000 sites where ASCL1 binds to, next column, at day 0, attack-seek negative sites, in other words, closed chromatin. And within 2 days, we see opening of that chromatin, and it stays open. So this is just one type of binding. We see different other types of binding where it binds and there's no change to the chromatin. And we see some where it binds and there's a reduction in chromatin binding, and some where it binds open chromatin and just sits there. So we've seen a diversity of these, but we're going to consider mainly the pioneered sites, if you will. So how does ASCL1 work? Well, as we showed previously, we curated out of the genome a site where ASCL1 would target that had eminase-resistant tags and was basically at a non-expressed site that could be activated, and captured 162 base pairs where we tried to have the ASCL1 sites as centrally as possible within the eminase-resistant and assembled it into nucleosomes. We made recombinant proteins as shown in the middle, and basically we're able to show using an electromobility shift assay that the combination of factors bound to the nucleosomes from the NR-CAM locus quite well. And so that's shown on the right-hand side. So we then actually took this sequence and dropped it in the middle of a nucleosome array, as Lisa Cirillo did so many years ago when we were originally studying FOXA1. And then you make it into nucleosomes from the free DNA, I should say. We make these NR-CAM arrays. We compact them with linker histone, being careful to use an equimolar amount of linker histone to the number of nucleosomes in the arrays. And then we add the transcription factor. And as you can see on the right-hand side, the red arrow, ASCL1 very robustly at the lowest concentration of 5 nanomolar, and this is against 13 nanomolar of nucleosomes in the reaction, you get a hypersensitive site. This is fully purified system, no ATP, no remodeler added, and so that's that. So given these capabilities, actually in this lecture series 5 years ago, I got a note from Yawen Bai, who said, let's collaborate and do some structural studies. So this message came through during the pandemic. And in fact, at the time, FedEx wouldn't even work for us to ship. So my grad students popped boxes of protein and nucleosomes in the car, and I personally drove it down to the NIH and dropped it off to Bing-Ri Zhu, who's a scientist working with Yawen Bai. So I acted as FedEx and then just drove back home, 3 hours there and back each way. So at any rate, lo and behold, not long after that, they had solved a cryo-EM structure, which showed a lot of asymmetry for the DNA that we had been assembling. That should say 162 base pairs, sorry, where there was an E-box motif near the dyad axis and then 2 other E-box motifs. And in fact, here's a movie, which is speculative, if you will, but this is summarizing what I'm going to show in more detail over the next few slides. And there's some conflicts. It's not perfect here, but this is essentially what we wind up with, which is that the binding of ASCL1 and E12A causes a marked shift of 15 base pairs of the DNA on the nucleosome as it binds. So remember, the starting nucleosome has the E-box motifs on the nucleosome, and it winds up like this. So this is a picture of what that final state would look like. And we've got 2 molecules of the ASCL1 E12A heterodimer bound as seen. So what's going on there? So we wanted additional evidence that there was actual movement upon binding. And so Bing-Ri Zhu developed a FRET assay where he put FRET donors at different places on the DNA and a FRET acceptor on the end of the DNA. So the donors are in the middle part of where the nucleosome would be. So at any rate, long story short, we saw a stepwise decline over time as we added ASCL1, a loss of FRET, a loss of fluorescence transfer, indicating that the Cy5 end of the DNA was being peeled off, if you will, as the DNA was rotating around the nucleosome. So we wanted to get at that in more detail, and Edgar Luzete Montero, who got this project off the ground and did the lion's share of the work I'm going to show, as a grad student, he did cross-linking of ASCL1 bound to the nucleosomes with DSBU with a 10 angstrom spacer, and then proteolysis mass spec. And lo and behold, got robust cross-linking to lysine 79 on the octamer surface in the nucleosome. And the main point is we didn't see exact physical contact between those domains. We had a linker and there was a gap there. So we wanted to test the functionality. This is the lysine 127 that was cross-linked and ASCL1, and it's at the beginning of helix 1 of the BHLH domain. And so we mutated that to 3 alanines because we figured, it might not be the lysine itself, could be the additional leucine valine, and we compared it to a bunch of BHLH factors. At any rate, this is what Edgar came up with, making a triple AAA mutant. So we expressed it in E. coli, as seen on the left, compared to the wild type, a fresh prep of wild type. And as you can see in the middle panel, the ASCL1 AAA mutant protein binds free DNA just as well as the wild type protein. But if you look on the right panel, it binds nucleosomes poorly. So the key point is we have a nucleosome-specific binding mutant of ASCL1. Well, we did some other assays, which was done by Marielle Corradin and Ben Garcia's lab, along with Edgar. And for that, we did hydrogen deuterium exchange, where we looked at the nucleosomes minus and plus ASCL1 E12A binding. And for this, you have your nucleosomes, and then you add deuterium, and you look over time at the extent to which the deuterium will exchange into the amide bonds of the proteins. And so it's, to some extent, it's like a protection experiment. Are you protecting the exchange, or are you eliciting a structural change in the protein that might affect that? It doesn't have to be protection. At any rate, we saw various sites of protection, and what I'm going to talk, or changes in deuterium exchange, and what I'm going to discuss is this one part where we saw exchange over the exchange perturbations over the acidic patch residues of the octamer. So the acidic patch was discovered by Carolyn Luger when she was studying LANA peptide binding. And after that, many other factors that bind nucleosomes have been seen to bind the acidic patch, and typically with what's called an arginine anchor, a bit of arginines that are closely spaced. So Edgar looked at the protein, and lo and behold, just upstream of the BHLH domain for ASCL1 is an arginine-rich domain with 5 arginines. So we made various mutants of this, and I'll discuss the all-Rs mutant, where we converted all of the 5 arginines to alanines. On the left side, you see again, we made the protein as shown. And then once again, we see binding to free DNA is perfectly okay with the all-R mutant, but binding to nucleosomes is greatly diminished. So that clearly said that we were able to get a different separation of function mutant in comparison to the AAA mutant shown on the right. If we put this in the FRET assay, most of the other mutants that we made and behaved like wild type, but the all-Rs mutant behaved a bit differently, being slower for changing the FRET parameters. So we also plug this into our nucleosome array assay, and the green arrow, shows that at 2.5 nanomolar wild type ASCL1, you get really nice chromatin opening in the assay, whereas with the AAA and the all-Rs mutant, it's deficient, and this is quantified from multiple experiments shown on the right. So these factors definitely have a nucleosome-specific binding defect. We then wanted to test their abilities and function in vivo, so Jingchao Zhang joined the project here, and he made a construct that co-expressed these two in mammalian cells. We plated them in primary human fibroblasts, not a tumor cell line, and looked at them at different times for chromatin status. Now we're just going to focus on the pioneered sites. There's nothing unexpected by the non-pioneered sites here. But at any rate, for the pioneered sites, this is now spreading the 18,000 peaks we had seen. And then you can see at day 0, they are TAC-seq resistant, and wild type, they get strongly opened. And we also see that they're enriching for MNase tags over the center of the binding events, showing that there indeed are nucleosomes there. And then when we test the mutants, you can see that the all Rs and the AAA mutants have a deficiency in the initial targeting by ChIP-seq. And this is reflected in an attack-seq profile where there's diminished attack-seq accessibility. So it seems that the primary defect of these is really just targeting the nucleosomal sites, analogous to what we had seen in vitro, and then the attack-seq effect seems to be likely downstream of that. And then finally, what I thought was super cool is that we asked if we could partition out the sites targeted preferentially by the AAA mutant or the all Rs mutant. And lo and behold, we were able to see some distinctions as seen here. So the middle arrow on the left shows that there are a bunch of sites that are more or less accessible by the AAA mutant than the all R mutant. And then going further down, there's many sites also that are a bit less accessible by the all R mutant than the AAA mutant, with consequent effects by attack-seq on the right. So the big question is, are these defective in cell fate control? And the answer is yes. The AAA mutant and the all R mutant, when you quantify on the right, they seem about 50% effective, but you can see by looking at the cells, they don't elicit the morphology change that we would like to see and seem to be clearly defective. So the big punchline from this whole talk is that nucleosome interactions by these transcription factors are important for efficient cell fate control. And the whole area of do pioneer factors exist? The fundamental property that we had initially described was nucleosome targeting, nucleosome scanning. And these data show that the physical interactions, as defined by biophysical and biochemical properties, are important for cell fate control. So thinking about the future, we want to better understand how this nucleosome modulation occurs, and maybe we can perturb that in ways that might be useful. And also, I just want to give a shout out to a paper that we just published in Science Advances by Andy Katznelson, where he made hybrid pioneer factors and was able to improve the targeting of heterochromatin, as I had described in the beginning as being a major challenge. So the work was done by Edgar Luzete Montero, Mei Lin, Fernandez Garcia, Marielle Corradin, Jingchao Zhang, Andy Katznelson, and Greg Donahue did a lot of work on the computational. None of this would have been possible without Bingri Zhu, and Yawen Bai, and Ben Garcia. Thank you for your time. I'm happy to take questions.
All right, great. Beautiful talk, and thanks for making up the time lost in technical issues. If anyone likes to, there was a hand by Jacques. Did we lose you? I'll start with a question while you guys come up with your own. As related to the site-specific targeting and the site-specific differences with the mutants, were you able to analyze those sequences and be able to make sense of, which DNA sequences were affected by those mutants?
So the differential targeting in vivo?
Yeah,
no, we didn't. I didn't show the data, but as best we can tell, they all have ASCL1 motifs. I don't think we've asked if they have subtle differences at one site versus another. My guess is that there'll be perhaps different chromatin modification states that are interacting with these different mutants, or larger chromatin fiber effects on access of those sites. But we think it'll be very interesting to figure that out in the context of what the genome epigenetically looks like where they are targeting.
Okay, thanks. All right, Rob, I think your hand is up.
Hi, Ken. That was really beautiful. I was just curious. So in binding to the nucleosome, do you think the DNA needs to breathe off the nucleosome for these guys to get access? Or do you think it's binding on top?
Right. Well, number one, there has to be something dynamic about the nucleosome for these factors to move it so much. There's no ATP, there's no work done. So they must be prevailing upon some intrinsic dynamic component. On the other hand, you can model these guys on a nucleosome, albeit transiently, without having to wrap their basic regions all the way around that might interact with the core histones. So that's kind of like the main parameter that we published in a 2019 molecular cell paper, where we screened 1,600 transcription factors, and the main correlation was whether they wrapped around the nucleosome or not. I'm sorry, wrapped around the DNA or not. And so, as best we can tell, they can do that, albeit transiently.
Right. Okay. It just seemed interesting that maybe if it breathed and then grabbed on, that would be a sort of a way of stripping the DNA as each one was binding, because the two sites were quite close to each other. So you can imagine it might add as a little lock to keep that DNA off the nucleosome, and then it's going to breathe further. I just wondered if that's the dynamics come from breathing and then binding to free DNA as opposed to transient interactions on top.
Well, I can tell you, I didn't discuss it, but we've mutated that site that's on the diet axis in the starting nucleosomes, the ASCL1 motif, and it has no effect on nucleosome binding, but it's important for free DNA binding. But mutating the two sites that were on the nucleosome and eventually get off, those sites are necessary for the transition. So somehow the factors recognizing those sites and eliciting it, and we don't see that final product in the nucleosomes without the transcription factors. So it's not like they're stabilizing some state that's transiently shifted like that, plus the FRET.
Right. Great. Thanks.
All right, Kai.
Yeah. Beautiful talk, Ken, as always. So the ChIP-seq, ATAC-seq were done at day 2. I just wonder, have you looked at very early time point, for example, if you could acutely induce a binding of ASCL1 and look at after 1 or 2 hours of induction, whether you also see reduced binding of this factor?
Yeah, that's an interesting question. We haven't taken approaches to do that. We figure by day 1 to 2, you're getting maximal induction of the proteins. We just haven't done that. It would be interesting. I should say, though, that Jacques Drouin's done that with PAX7, which targets closed chromatin and elicits ATAC-seq sensitivity. And he did these careful time courses with an estrogen fusion to PAX7 and basically was able to clearly show initial binding of the transcription factor prior to the chromatin opening step. And then some other people have done similar things. So my guess is we would just see that initial targeting to be defective with the AAA and all Rs mutant.
Thank you.
Okay, Jacques is back. So would you like to ask your question?
Yes, I'm back. Sorry about that. Beautiful talk as usual. It's always so exciting and impressive to look at all this. I just had a question because I just read a paper recently about the ACL1 in neuron differentiation being linked to H2AZ. Do you think that if you would use an H2AZ nucleosome, you would see even better dependency on acidic patch because the acidic patch is larger in H2AZ nucleosome?
Yeah, that's an interesting question. We hadn't thought about that. It's certainly something that could be done. We still haven't shown a direct physical interaction. It's inferred from the data. And going back to Rob's question, when we have the structure of ASCL1, the two heterodimers bound to the nucleosome, we don't see protection of the acidic patch in that. And it could be transient from some other part of the protein. Another possibility is that that's one of the ways that the factor initially interacts with the nucleosome when the DNA is on the nucleosome. And then as things progress, it will eventually release that interaction.
Yeah, sounds good. Would be interesting to look at the affinity.
Yeah, thank you.
Thank you.
Okay. And for our last question, Sihem, Do you want
Hi,
to?
Ken. Very nice work. I was wondering how specific so the lysine and the arginines that you've identified to ACL1, and could this be kind of more generalized features to other pioneer factors?
Right, good question. There have been some papers, some structure papers, and I should have mentioned this. Actually, it was on the slide. There's one or two other transcription factors. I think GATA4 and perhaps FOXA that have been studied, and people have seen interactions with the acidic patch. Most of the acidic patch interactions have been non-specific DNA binding factors or just chromatin associated factors that interact with the acidic patch. So it's kind of a new era. And I think it's certainly worth looking at your various factors if you have an arginine-rich domain that could be close to the DNA binding domain. We did that with ASCL1 in comparison to some of the other BHLH factors, and it was not highly conserved. So we think that it evolves specially perhaps for these factors that are so potent in cell reprogramming.
Very interesting. Thank you so much.
Okay,
I'd
great.
like
Thanks
to
for the discussion.
thank both speakers for the wonderful talks and Alyssa for co-hosting with me. So this first session of the ASBMB transcription webinars have been a great success. So look forward to more sessions in the future.